So I'm really happy to see more and more people joining. But first of all, of course, I want to also thank our today's speakers. And we're going to start now basically with two of them, so which will be Miranda and Sarah. And they will talk about, so they are among four, I think if I remember correctly, four co-authors, first co-authors, and of the recent Nature paper entitled Single-Cell Spatial Immune Landscapes of Primary and Metastatic Brain Tumors. And just briefly for, so Miranda is a PhD student in Daniela Quayle's lab in the McGill University, and Sarah is an MD PhD candidate, right? Finishing her PhD in the lab of Peter Siegel, also at McGill, and the institute is located in Montreal. And we will have a talk from both of you. And super excited and looking forward, and thanks again for the accepting our invitation here. And yeah, with that, I think we can start right away.
Wonderful. Well, thank you so much for the introduction, Michael. Let me just share my screen here and get a laser pointer going. There we are. And with that, it is our pleasure to speak with you all today about our paper that was published earlier this year, as Michael mentioned, in Nature. So Miranda and I wanted to take this opportunity of this talk to describe not only the data of this paper, but also really emphasize the project as a whole, including how exactly it got started. So as Michael mentioned, my PhD is under the supervision of Dr. Peter Siegel and is focused on brain metastases, specifically microenvironmental mediators regulating how brain metastases grow once they become established in the brain. And to do so, our lab houses a large brain metastasis patient tissue biobank, which was spearheaded by Matthew Dankner, a previous MD PhD student in the lab. And we developed an interest in assessing the tumor immune microenvironment of our patient samples. So we reached out to a lab conveniently down the hall who has expertise specifically in the tumor microenvironment.
Yes, and that's where we come in. So we were the lab down the hall. I'm supervised by Dr. Daniela Quayle. And like Sarah mentioned, our lab is particularly interested in the tumor immune microenvironment of many different cancers. In my project, I have a particular interest in glioblastoma. And so our two labs really had this common interest and goal of characterizing the brain tumor microenvironment. And that really led us to join forces on this big project. So in our lab, we spent some time building these large TMAs with glioblastoma patient tissues. And that would allow us to profile these tumors alongside all of the metastasis tissues that were banked by Dr. Siegel's lab.
So we wanted to take a global look at many of the different cell types of the tumor microenvironment, both immune and otherwise. So we turned to the novel, highly multiplexed imaging mass cytometry system. So unlike standard diagnostic histology that is limited to one marker at a time or up to 5 markers through immunohistofluorescence, where you're then limited by fluorophore wavelengths, the imaging mass cytometry system instead lets you use up to 40 markers simultaneously on a single tissue section. And this not only lets you visualize many cell types all at once, it also maintains tissue architecture to provide spatial information for each cell. And this is something that is normally lost through other single cell technologies that instead rely on dissociated tissues. So we leveraged this technology on a combined cohort of glioma and brain metastasis patient samples. And between the biobanks of our two labs, we profiled more than 1.1 million cells across the tumors of 139 glioma patients and 46 brain metastasis patients. And our cohort included brain metastases from different primary sites, predominantly lung, melanoma, and breast, as well as looked at two specific regions of the brain metastases, either the center of the metastatic lesion, which we term the core, or at that brain tumor interface, which we term the margin.
So as Sarah mentioned, we used IMC because it allows us to look at a large number of markers within a single tissue. And we can do this because it takes advantage of metal conjugated antibodies that are detected by cytometry by time of flight, rather than your traditional antibodies that are conjugated to fluorophores. So this really gave us and allowed us to get a big picture of the tumor microenvironment and start to map where these cells are and how they're really interacting within the actual tumors. So to be able to analyze this large amount of data, we partnered with computational experts in Dr. Logan Walsh's lab, particularly Elhem and Morteza. They are the computational masterminds who really developed the segmentation pipeline that we used in our paper, and also drove a lot of the spatial analysis that we'll present later on. And just to give them a shout out, they also led another project in parallel to ours, looking at lung cancer specifically. And they took a bit of a different approach with this paper and applied machine learning. That was super cool. It was published at the same time in Nature. So I really encourage everyone to check that out if they're interested. So for our study, we optimized a panel of antibodies that allowed us to identify 20 unique cell types within the tumor microenvironment. So this included a number of major immune lineages, as well as structural cell types like the endothelial cells, the astrocytes, as well as the cancer cells themselves. There was a small fraction of cells that were not captured by our data and by our panel, and we called those our undefined cells throughout our data.
So given the time constraints of this talk, we understandably can't go through all of the nitty-gritty data that's in the manuscript itself. So instead, we will be staying relatively high level and highlight some of the different types of information that can be gleaned from the IMC dataset, such as ours. So these big picture kind of analyses can include higher level cell population dynamics or frequency analyses, as well as higher order spatial analyses, such as pairwise cell interactions or multicellular neighborhood analyses. So starting off with cell population dynamics, these are types of analyses that are particularly useful as a benchmark against what is known in the literature to serve as a form of quality assurance for lineage assignment and your cell segmentation pipelines. So these analyses enable you to characterize the frequency of cell types across specific cohorts to look for some expected patterns, for example. Here we show that broadly gliomas were immune cell deserts compared to brain metastases, and instead were enriched in astrocyte components, shown by these beige bars here. As well, in the brain metastases, we see that it's the melanoma brain mets that are lymphoid rich compared to tumors that originate from other primary sites. And this is especially apparent when you look at those brain met margins. And these analyses can go further when you query specific clinical parameters, as shown by our bubble plot here, comparing frequencies across many of our different clinical groups. For example, we observed elevated frequencies of various microglia populations in our brain metastasis patients without leptomeningeal disease. And for gliomas, we saw distinct differences in frequencies between IDH wild type and IDH mutant gliomas, such as higher frequencies of these NK cells in the IDH mutant tumors. And this is why it was very important for us to define glioblastomas by the most updated WHO guidelines, which only classify IDH wild type tumors as glioblastomas. The IDH wild type and mutant tumors really seemed to be distinct in terms of their pathology and clinical outcomes, as well as their immune composition. So this is something we kept in mind throughout the paper. But as I mentioned before, much of the power of IMC technology lies in its spatial resolution. And one type of spatial analysis that we performed was this assessment of pairwise cellular interactions. So we leveraged the methodology that was first developed by the Bowden Miller lab in their 2017 Shapiro et al. Publication. and updated it with a new method to quantify the likelihood of interaction and avoidance between individual cells. And shown here is a heat map depicting the average pairwise interaction avoidance scores across cell types in our glioblastoma, brain met cores, and brain met margins, shown by the different rows in each square, with the tendency to interact shown in red and the tendency to avoid shown in blue. And while there's a lot of data you can take away from this heat map, one example that we can quickly walk through is this idea that cytotoxic T cells largely interact with cancer cells, shown by the red color of the box, but those interactions are generally greater in the brain metastasis cores compared to the glioblastomas, shown by that darker middle row. And the spatial information can be leveraged in even higher order analyses, such as multicellular neighborhoods. So we used the approach originally developed by Gary Nolan's group in their 2020 Shirk et al. Paper. And here we took each individual cell and then looked at the 10 nearest neighbors to that cell. And based on the identity of those 10 neighbors, that central cell can be binned as being part of a cellular neighborhood. So we identified 9 neighborhoods that were common across brain tumors, both glioblastoma and brain metastases, with the cellular composition of each neighborhood shown in this heat map here. And what was interesting is that our cellular neighborhoods did recapitulate known tissue features, including the tumor boundary and tumor compartment, the vascular niche, as well as regions that were enriched in astrocytes as well as macrophages. And when you look at the distribution of cellular neighborhood abundance in samples across clinical subgroups, you start to see distinct differences between glioblastoma and our brain mets, for example. Specifically, that our glioblastomas are dominated by cellular neighborhoods that are enriched in astrocytes, while the brain met cores are dominated by the tumor compartment cellular neighborhood, which reflects this infiltrative nature of gliomas compared to metastatic tumors. And moreover, we found that neighborhood similarities in brain metastases were dictated regionally rather than by primary tumor type. So the margins, irrespective of primary type, more closely resembled the cellular neighborhoods of the glioblastoma samples compared to the brain metastasis core samples.
So now that we've defined these neighborhoods, we wanted to know whether this spatial information could be clinically meaningful. So in brain met patients, unfortunately, it's really challenging to assess survival outcomes because these are often late stage patients. They have a ton of extracranial disease, and that can all confound the survival info that we have. And so we turned to glioblastoma, where we were lucky to have access to a cohort of long-term survivors. And we decided to assemble a clinically controlled cohort of patients. This was really important because we know there are a number of clinical factors that can influence a patient's survival. In particular, the level of resection that they get at the time of surgery will really influence how long they will survive. We also know, as Sarah mentioned, that IDH status is really critical to the definition of glioblastoma. And also MGMT status will influence how these patients will respond to treatment. So we balanced and controlled for all these things. And in the end, we had 16 long-term survivor patients. So these are patients that lived beyond 3 years. And then we also had 16 short-term survivor patients. So patients that survived less than 1 year. And interestingly, when we looked at the neighborhood composition across these patients, there was really only one neighborhood that was significantly enriched in long-term survivors, and that was the macrophage enriched neighborhood 7. So we were a little surprised by this because macrophages have broadly been associated with poor outcomes in glioblastoma, but we're beginning to appreciate that these cells are far more heterogeneous in the tumor than we once thought. And so we were really interested in taking a deeper look at the macrophage compartment to try to figure out what was going on. So to do this, we performed unsupervised clustering of all the macrophages and monocytes across all of our glioblastoma images using the functional markers we had in our panel. We saw that there were these clusters that were enriched in long-term survivors and not really there in short-term survivors. And interestingly, these macrophages were defined by their expression of myeloperoxidase, or MPO. So again, we were a little surprised because we typically find MPO in neutrophils. We know it's an important component of a neutrophil's innate defense system against pathogens, and it's been much less described in macrophages. But we did confirm by immunofluorescence staining that these were macrophages expressing MPO rather than neutrophils. We looked into the ontogeny of these macrophages. So in brain tumors, macrophages can either be yolk sac-derived microglia, so these are the tissue-resident macrophages of the brain, or they can be monocyte-derived macrophages that will seed into the tumors from the bone marrow. And we were able to distinguish these populations using P2Y12. When we looked at the distribution of the MPO macrophages, we really saw that they were predominantly monocyte-derived, so they're coming in from the periphery, and they also lacked the scavenger receptor CD163. To understand how these cells could be functionally different, we probed 3 publicly available single-cell RNA-seq datasets and performed ingenuity pathway analysis on the differentially expressed genes that were common across these 3 datasets. And we found that MPO macrophages had these enhanced cytotoxic pathways. So they had upregulation of phagosome formation, as well as ROS production, and so on. And not only were they enriched in long-term survivors, we also saw a significant reduction in the recurrent tumors, even compared to primary tumors of short-term survivors. So this really suggested to us that these cells diminish with progression and could have some sort of role in controlling the tumor. And finally, we wanted to see if MPO macrophages could be indicative of better outcomes using immunofluorescence staining, because it's a more feasible approach compared to IMC, and it's already being widely used in the clinic. And so to do this, we used an expanded cohort. We used a subset of our existing patients just based on tissue availability. And then we had a number of samples coming in from the University of Calgary, as well as the Netherlands Cancer Institute. And we stained these tissues with MPO and IBA1 to track the MPO macrophages. When we ranked patients based on the frequency of MPO macrophages, we did indeed see that these cells were associated with better overall survival. So we're now doing some follow-up work to try to understand what exactly these cells are doing and how we could potentially therapeutically induce this population. We also have some follow-up projects on how the microenvironment is related to response to therapy, and that includes immunotherapy and radiation. So we have some samples from Checkmate 498 as well as Checkmate 548, which were clinical trials testing anti-PD-1 for glioblastoma. And we're also interested in the POBIG trial as well, which is testing preoperative radiation. So for this project, we're collaborating with Dr. Leila Akari at the NKI. Their group just published this wonderful study on radioimmunotherapy resistance in glioblastoma, and I think everyone should check that out.
And on the brain metastasis side, our ongoing work looks to further characterize the tumor microenvironment between clinical subgroups of brain metastases. So our lab has interest specifically in histopathological subtypes of brain mets. We have previously shown that brain mets can grow in 2 distinct histopathological patterns in the brain, either as this minimally invasive lesion with this sharp demarcation between the tumor and the surrounding brain, or as this highly invasive lesion with tumor cells outwardly invading into the distant brain parenchyma. And we're currently looking into the microenvironmental differences between these 2 distinct tumor types. And with that, we would like to acknowledge all of our collaborators, both from McGill and the Goodman Cancer Institute, as well as abroad. As we alluded to earlier, this was a very large collaborative project with many different people involved. And we specifically want to highlight the core facilities at the Goodman Cancer Institute, especially the single-cell imaging and mass cytometry analysis platform, which allows us to perform this IMC in-house, as well as have our own optimized panel of antibodies of interest. And with that, thank you very much, and we would be happy to take any questions.
Cool. Thank you so much, Sarah and Miranda. That was really great talk and nice overview about the paper. And yeah, now it's time for the first round of discussion and questions. So whoever has questions, just raise your hand and unmute yourself, or rather type them in the chat and we can just read them. And until first questions will come in. So I think I may start. Oh, no, sorry, I will hand over to Angel. I just saw that he also has a question. Please.
Yeah, thank you. Very, very nice talk, very nice walk. So I was wondering whether there is any kind of readout that you can measure in the periphery to try to identify either this neighborhood which you have correlated with better survival, or with the presence of MPO positive macrophages, thinking in the long term, if you are planning to somehow treat these tumors to try to increase the proportion of this population, which might be beneficial, how would you segregate the patients pre-surgery, if that's something that you have thought about?
That's a really good question. So that's exactly what we're looking into now. We're really trying to figure out what are the precursor populations that are becoming MPO macrophages in the tumor. So I don't have the answers right now, but we're definitely looking into it because that'll be important down the line in the clinic for sure.
Okay, thanks.
Yeah.
Okay, I will briefly take another question now. So as far as I remember it correctly, there was also a point in the paper where you showed basically that there is a higher endothelial frequency, which was associated to long-term survivors, as far as I remember it correctly. So was there any kind of also correlation, like basically to the location per se of the tumor within the brain? So because of course it could be that there is the location per se, like of the developing tumor, like maybe influencing also the frequency of the vasculature and of the endothelial cells.
Right. So on the brain metastasis side, we did use that heat map I showed, looking at cellular interactions. And some of the interesting ones that were jumping out were these endothelial cells and how they interacted specifically with the cancer cells themselves. And in addition to all of the lineage markers that Miranda described, we also had a handful of functional markers on top of that. This included Claudin-5, which we used as a marker for endothelial vasculature intactness and junctional markers. So we were able to see that our endothelial cells, there was difference in expression of this Claudin-5 marker, lower expression indicating leakier vasculature. And we were able to identify these peritumoral vasculature, these peritumoral endothelial cells were leakier, and that was able to associate with some of the edema data that we do have, which again fits this idea of just leakier vessels altogether.
Mm-hmm. Yeah.
And Michael, sorry, were you also asking about the endothelial phenotype in the long-term survivors of glioblastoma?
Exactly. Yeah. Yeah. So,
okay.
Yeah, because I think as far as I remember correctly, that you also described that there was like in this long-term survivor group, there was like also higher frequency of endothelial cells per se. So this of course might be that it's basically related to the like location within the brain itself, right?
Yeah, it's very possible. We actually took multiple samples. So we sampled multiple tumor regions for many of the patients for that exact reason, because if you're just sampling one region of the tumor, of course, like these tumors are very heterogeneous. And so we were trying to capture multiple regions. It's possible that the endothelial cell frequency could be related to chemotherapy delivery. That's one hypothesis. But yeah, that was an interesting finding for sure.
Yeah, thanks a lot. Any further questions for the first half of our seminar today? Until now, I don't see anyone raising hands. So I would suggest if there are some more questions coming to your mind, just write them in the chat, and then I think we will move on with the second talk of today.